So what you're looking over here, the white stretch is your fragment. So you have chopped off your DNA, whatever input DNA you wanted or RNA you wanted, and you have chopped off that into subsequences or fragments as we call them, and you have added these sort of adapters, which they call the one in the green, the red, and the other colors. So what they would do is they would help these fragments to go and bind to the surface of the machine, so which you will see in the next slide. So these are pre-decided, these adapters, and what you can do is, because of these adapters and also something called as indices, you are now able to carry out the sequencing of multiple samples. So let's say you had 10 wild types and 10 mutants, and you want to run the samples together. Because of these addition of these adapters, this was now possible. One second.
Hey, Mohak?
Yeah.
I have a question. When they chop up the sample, they use some generic endonuclease or something that cuts all over the genome.
Mostly they use sonication, and then they do a size selection based on whatever fragments you get.
Oh, okay. So then, but to get the adapter molecule to ligate to the fragment, then
Yeah.
post sonication, do they generate? How it generates sticky ends also or something? I don't know.
So they generate what?
No, because to get the adapter molecule to ligate to the fragment, you need some sticky end on the fragment, right?
You need what? Sorry, I'm not able to hear
that. Like a overhanging bit.
Yes. So so there are multiple steps during a library formation. So obviously when you do a fragmentation, you might end up with different moieties. So then you do an end repair. After doing an end repair, you usually have this. One of the techniques that I am aware of is they have this TA overhang technique. So where they specifically add a particular overhang at the end of these repaired ends, and then after that, once you get an overhang, you can then use whatever adapters you want to use.
Okay. No, I understand. Thank you.
Yeah, so this part is the last part, which is the analysis. So a couple of things to note over here. The first is when they were talking about these clusters, each cluster would be equal to one sort of read. So within a cluster, there will be a number of reads. That is, you're amplifying these reads. So one cluster represents one read. So in a file later on, when we will see these reads actually were a part of one cluster, and so on and so forth. So that was the basic thing. So cluster would equate to one read, and then you amplify that particular read, and you get more copies of that molecule. So any doubts so far? Hello.
No doubts.
Okay. So also one more point to note over here, that with addition of each nucleotide, there was a fluorescence um, that was emitted when that particular nucleotide was added. So this is the fluorescence that is noted down by a camera, and that is what you were also seeing in one of the visuals, where there were uh, a lot of colorful spots that were coming up when you look at from the transverse section. So so that's how the whole sequencing happens. So now, because of this, it's a massively parallel sequencing. You are now able to amplify a lot of DNA, get a large amount of your initial sample. Not just that, even a large number of samples can be sequenced at the same time. And with time, so it started in 2000s, early 2000s, where so this what you're seeing over here is NIH has provided this cost per human genome. So it started with $100 million per genome sequencing of a human, but in today's date, it has now come down to around $1K per sample. Um, and this has sort of now stabilized for the past 5 years. So definitely the cost has come down, and now imagine doing the same thing with Sanger. It would have been a much higher cost and much more laborious as compared to next generation sequencing, which we now know of. Then a number of things then became even more possible. So remember, we talked about certain questions that we could target via Sanger sequencing. But one of the shortcomings of Sanger sequencing was that cross-species comparison or within species comparison was not possible using Sanger sequencing, just because of one of the reasons being low throughput. Now, because of next generation sequencing, you can do things like RNA-seq or gene expression studies or SNP analysis, de novo genome assembly, and so on and so forth, because you get large number of short reads. So these reads are usually up to 250 base pairs and so on in length. And you will get millions of reads. So for example, for an E. coli genome, which is 4.6 million base pairs, you would get you can get up to even 10 million reads, which is a great read number of reads which you can get for a 4.6 million base pair genome of E. coli. So then there are still efforts which are going on in this direction. Then we have third generation sequencing, which is nanopore and SMRT, which is single molecule real-time sequencing, where you don't need to amplify at all. You are using things like pores and polymerases to just take the complete DNA at a stretch and just keep adding, keep noting down what those nucleotides in that particular molecule are without even amplification or fragmentation. So because of this, this um, um, function of third generation sequencing, de novo genome assembly, we can achieve a good de novo genome assembly because this gives you longer reads. So up to 10 kbs of reads can be obtained by third generation sequencers, as opposed to the next generation sequencers, which can only give you very short reads up to 250 base pairs. Now, there are pros and cons to both of these things, which we will discuss as and when we talk about when we talk about other things related to NGS. So these are the two videos that I found are really nice. So you can go back and look at these videos. They explain beautifully how nanopore and SMRT work. Yeah. So then the question really is, which of these three sequencing approaches would be most suitable for RNA-seq based differential gene expression studies across samples, which we are going to talk about in the coming 2-3 lectures. So hold this question for now. We will come back to this at the end of this talk. Then probably we'll be able to say which of these three techniques are really most useful for doing gene expression based studies. Then the next part is, now we are going to talk about one of these algorithms which is very fundamental in analysis related to NGS outputs, which is the read mapping problem. So DNA is a string really, when we talk in terms of computational aspects, and you can get a DNA in a form of a file. So this, what you're seeing over here, is a part of Escherichia coli genome. And what sequencers do is they, as we already discussed, they will chop off this DNA and they will create fragments of varying sizes.
Mohak, you got stuck in between.
Where did I get stuck?
This previous slide, when you were introducing this algorithm, like it's a problem, like.
Oh, this one, the contents?
Yeah. So when you were saying read mapping problem, that's it. Like that was the last thing that we could hear. Yeah.
Okay. Thanks. So the next section we are going to talk about the read mapping problem, which is one of the fundamental problems in the analysis related to next generation sequencing. And we will come to that, what this problem is and how have people tackled this problem. So now we are going to shift gears from talking about the biology of the DNA to how we can computationally look at the DNA. So in that sense, one of the first things that comes into mind is that DNA can be written down in a file as a big string. So what you're seeing over here is Escherichia coli, a part of the genome of Escherichia coli. And what sequencers do is they will chop off this DNA. So if let's say you give this as an input to the sequencer, it would chop off this DNA in fragments of varying lengths, and then gives those read out reads or fragments in the form of a file, which then finally you analyze. So if you remember, 2 months back, we had done a de novo genome assembly already there, where there was no reference to begin with. There were only these sort of files where you just had these files and you had to blindly go back and try to see what that big sequence would be. So that is what we call as the de novo genome assembly, where you are moving from the right to the left, where you do not have this left with you at all. But uh, that's the first thing. Now, usually people have been sequencing uh, and getting the assemblies of different sort of DNAs of different species uh, every now and then. So, for example, you have Escherichia coli assembled genome present on the database. Now, let's say you do want to do a study where you want to see if, given certain conditions, how does the genome change, or how do the expressions of these the genomes change. Now, because you already have the assembled genome with you, you need not have to do the assembly of each of those samples that you carry out in your experiments again and again, because de novo assembly is a cumbersome process, not just in terms of the time, but also in terms of the optimizations and in terms of achieving the best possible assembly um, that there is. So over there, read mapping problems come handy. So given that you already have a genome available and you want to carry out a large amount of study related to a genome whose DNA you already have assembled, then you can save on the time and even accuracy when you do the read mapping of these reads against a reference, as opposed to assembling each of these sample files and then proceeding with the analysis. So, so read mapping problem is basically a string search problem if you talk in terms of computer science. So what you have is you have two inputs. Let's say you have a genome, which is this that I mentioned over here, and you have a query. In this case, it's 80GGG. And all you want to do is you want to see that, okay, tell me where is my read coming from in a given set of genome. So that is all the exercise is. So the first thing that comes to a layperson's mind or somebody's mind is that, okay, the easiest thing would be a brute force approach, that I take this read and I start from the first position of the genome and keep seeing that, okay, am I getting this read a complete match or not? So the best case scenario is that yes, as soon as you start doing the search, you will get the match at the first position of the genome. So you already have a match. This is the best case scenario. But let's say now I change the read. Now the read is AGGTA, which is not, if you align this to the first position of the genome, you will not be able to get this particular read. So what you will then do is, you will start shifting this read and start looking at other positions in the genome. So let's say now this was the first genome, there was a mismatch. First position of the genome, there was a mismatch. Then you try the second position of the genome, there's again a mismatch. Third, again a mismatch, and you will keep repeating this exercise till the time, if at all, you get a match. So this is the worst case scenario, that the read would only match to the end of the genome. Now remember, if you talk about a human genome, which is 3.1 billion base pairs, or a Escherichia coli genome, which is 4.6 million base pairs, doing this exercise for this read would be going to take a large amount of time. Are you guys with me so far?
Yeah.
Yeah. So then a typical bacterial reads file, which is usually 150 to 200 base pairs, would contain more than 1 million reads. So imagine doing this exercise for all 1 million reads. Let's say you have 1 million reads. If you do this exercise for all 1 million reads, this is going to take a large amount of time. It is not going to be practical. And usually how we report such time is a big O notation. So that means the amount of time it will take will be a function of the length of the genome and the combined length of all the patterns or all the reads in this case. So 1 million into, let's say if the one read is 150, so 1 million into 150 into the size of the genome is the amount of time that is going to take for a search or searches of these reads against these genomes. So definitely brute force approach is not going to happen or not going to work when you have large data. So what can then be the solution? So one of the things that one can think is, now I will give you an analogy. I'll try to give you an analogy for this. Now remember that you want to buy certain items from a grocery store, and if what you do is you go to the grocery store one at a time. So you go to the grocery store, ask the person over there that, do you have item A? The person tells you it is there or not. You come back home, then you, because you are not able to for some reason remember the whole list, you are going back and forth to the grocery store and trying to see if you have that item or not in the grocery shop. uh, that is what the brute force approach was. Now the, or what you can do is you can write down each and every item that you want to buy in a list, and that is what we also call as a data structure. You will now take the grocery list all together to the shop, and at one time, in one go, you will ask the owner over there that do you have item A, item B, item C. So this will save you a lot of time in going back and forth. So grocery list is the simplest form of data structure, and that is what people then realized would be used for even read mapping over here. So let's try to see what data structure that would we come up with. So now let's say we have these bunch of reads now, AGGT, triple GC, ATGG, and so on. And we are going to use a data structure called as a trie. So now let's remove the genome for now. So we are going to make a grocery list out of these reads as of now. So how we make a data structure out of this is we start at the root and we take the first read and we start building up the read in the form of edges and nodes. So each edge would be one nucleotide, and as you move down a particular path, it will give you the length of the particular read. So in this case, the first read over here would be A, G, G, E, and all the nodes would be the cumulative of the nucleotide so far. So this node would be A, this node would be AG, this node would be AGG, this node would be AGGT. So this is how you would get to this. Now, when you move to the next read over here, which is triple GC, now from the root, there is no G already available. So you will start off a new path, which is GGGC. And then when you come to the third read, you already have an A next to the root. So now what you will do is you will go to this A. Now from A, next is not a G again. So it is a T. So you will bifurcate it to the other direction, and you will again go to A, T, G, G. And similarly for the last read, you already had a G next to the root. You will then only bifurcate from the after the first node, which starts with the G. Are you guys with me for so far?
Yes.
Yeah. So now we have this data structure, which is a trie, and that is what you can analogously say is a grocery list in this sense. Now, what would happen is rather than taking each read, now you are going to shift, or you're going to take this whole data structure at one time to the genome and carry out the same thing that we did for the brute force. So what it would mean is, let's say I take my data structure to the first position of the genome, and I see, is there a A? Do I see a A in my data structure? Yes, I see. Then next, do I see a T? Yes, I see a T. Do I see a G? Yes, I see a G. Do I see a G? Yes, I see a G. And now we have reached the end of this particular path. So, and we have got a complete match. So we can say that yes, there is a read that is mapping to this particular position in the genome, which is the 0th position if we start counting from 0. So now remember, because of this, this would only take the amount of time which will be proportional to just the length of the genome and the biggest query length in your data structure, which will be much less as compared to the brute force approach. So definitely try is a much better mapping approach than brute force. But now remember, we fragment our DNA into a number of chunks. And because of this, usually you tend to get a large number of reads per sample as opposed to the length of the genome. So for example, if your E. coli genome is 4.6 million base pairs, usually you would want to get number of reads which would be up to tens of millions of reads per sample. So therefore, the number of reads will be much more as compared to the length of the genome. So now you can imagine that this technique is still going to take a large amount of time. So then we can ask, can we still do better? The answer is yes. What we can do is, because your length of the genome is less as compared to the number of reads, how about we make a data structure out of the genome itself and not the reads? So we can pre-process the genome instead of all the reads into the data structure. So what we call that thing is a suffix tree, and it's also called as an offline approach. So whenever you pre-process a genome, it is called as an offline approach. So let's say now we take a genome that is banana and we end it with a end character. So backslash n is nothing but it demarcates that, okay, now the genome is going to end. So that is all that is to the backslash n. So if we make a try out of banana, uh, what we would want to first do is get the list of all the suffixes that are possible. So you're going to get a banana, you're going to get an anana, nana, ana, na, and a. And finally, end character to just demarcate that this is the end of the string. So now we will start arranging these suffixes. So suffixes, when you start reading from left to right, that is a suffix. Now I will go, so we are going to repeat the same exercise of making a try, but this time with all these suffixes, and you will get a structure like this. And definitely this will be much more handy as compared to making a try out of all the patterns or all the reads of your sequencer. Then what you can also do is you can, hello.
Yeah.
Are you guys able to hear? Okay.
Yeah.
So then you can merge these. So let's say now the example of this banana node, which is this path, which is spelling out banana. Because there are no bifurcations in this particular node, we can merge this together and write this as one node as banana over here, right? And you can do the same exercise wherever you are not seeing any bifurcations. So there is no bifurcation at this place, so you can merge this together. So what would happen now is you're merging these suffixes, and this is what we call as a suffix tree. And the leaves over here, which are the squares, will tell you the index of that particular suffix. So banana will start with the 0th index, 0, 1, 2. So banana will be 0, anana will be 1, nana will be 2, ana will be 3, and so on. So this is what you will mark over here. And then you can store this suffix tree, or you can store it in the form of an array. You can remove this tree altogether, but you can just note down these suffixes and set them in an alphabetical order or a lexicographical order, and you can also note down their indices along with them. So now imagine not storing this, but only storing this. So this is going to save you a large amount of space and time altogether. So if we do an analysis on the time and the space that it would take, if we talk about the suffix tree, it is going to take the time that it is going to take will be proportional to the length of the genome, also the length of the query. And let's say if you store this, let's say I tell you that this is taking 50 GB of space, a suffix array would only take 15 GB of space in this case. And the search would also become very easy. It will be a binary search. So for example, I tell you that my read is anana. So you don't have to now look at 0, 4, and 2. You will bifurcate this array into half, which is a binary search, and you will only look at anything which is before A. So A, anana, ana, A, and dollar. You will not be going to B and so on, because now you know that your read starts with an A. So it would be a futile thing to look at read starting with B or suffixes starting with B and N. Is the concept of suffix clear to everybody? Because this is going to be crucial for the next thing that we are going to do. Is suffix clear?
Yes.
It's clear as of now.
Okay. So definitely the offline approaches, when I say offline, I mean where the genome is pre-processed, are better than the online approaches, which is when the genome is not processed. And so far what we have seen is that the worst is the brute force and the best is the suffix array. But could we do better? That is still a question, because we will be dealing with a large amount of data and we want to scale our algorithms and not compromising on the time and space. Can we still do better? And the answer is yes. We will be now talking about Burrows-Wheeler transform, which is one of the most widely accepted and used alignment, read alignment or read mapping aligners that exists. So Burrows-Wheeler transform is not new to the field. Historically speaking, whatever compression that you do, so you do a dot zip, gzip and so on, there is a particular kind of compression, which is bzip, which can compress the data a lot. So over there, we use this Burrow-Wheeler transform to do the compression. So we will start looking at what a Burrow-Wheeler transform is, and then we will finally get to how the alignments are done with respect to Burrow-Wheeler transform. So now let's say you have a database genome, which is ACAACG dollar. Now again, dollar is the end of character, end of string that I am assigning. It is not the part of the genome. This we are assigning on our own, just to say that, okay, this is where the genome ends, a dollar sign. And the aim is to find the Burrow-Wheeler transform of this particular genome. So how we go along doing that is, you start with a string and you look at all the circular permutations that are possible. So what I will do is, I will shift this dollar to the first position. So I will get a dollar ACAACG. Then now this G, I'm going to shift it cyclically to the beginning of the string. So I'm going to get G dollar ACAAC, and I will repeat the exercise till I get the dollar from the beginning till the end. Are you guys with me so far?
Can you repeat this once?
Yes. So we are going to do circular permutations. So if you start with a string, I will be permuting my string in all the possible combinations. So when I say a circle, what I mean is that my the last column over here is directly linked to the first column over here. So therefore, there is no sequence as such. And because of this, I will be able to keep shifting my strings from the last to the first. So now if you look at the first row over here, what I have done with respect to this string is, I have taken this dollar and I have put it in the beginning, and I have shifted everything with one space or one position. So the total length remains the same. I'm just shifting everything by one position. So my dollar, which was at the last over here, it comes to the beginning. Now G becomes the last. Next iteration, I will put my G in the beginning. Now C becomes last. I will put it again in the beginning, and I will repeat this exercise till the time I have shifted everything which was there initially in the string to the first position. Is it clear?
Yeah, thank you.
Yeah. So now what I have highlighted over here is the one in the bolds, which are ending with dollars. If you now notice or look carefully, these are nothing but the suffixes of this particular string. So in a way, the Burrows-Wheeler transform uses suffixes in computing whatever it wants to compute. So I mean, you can see that ACAACG is a suffix, the largest suffix. Then CAACG over here is again one of the suffixes till the time you reach this suffix. Then what we are going to do is, we are going to sort them in an alphabetical or a lexicographical order. And now an important thing to note over here is that dollar has the least sorting value as compared to the other characters. So because of that, dollar will always be the beginning. So you will get, this particular string will remain the same. Now the next would be AAC. If you look at up in like a telephone directory, from all of these strings, this particular string AACG would come next, ACA would come next, ACG would come next, and then you enter the C's, and then you enter the G's. Is this clear? The lexicographical sorting?
Yeah, so I have one query.
Yeah.
So when reads are like quite long, this lexicographic sorting, will it not take time, like, and memory if we have used data as compared to binary sorting or something like that, number sorting?
So yes, so the thing is that once you create an index, so we are basically creating an index. Once you create an index, it is also not just creating something, but it is also when you are searching something. So the searches are going to take a much lesser amount of time as compared to all of the previous techniques that we have talked so far, which we will get to in the next slides. So it is not just making the data. So for example, if you have already made a database, so for example, if you make a human genome database, you can always download the human genome indexed sequence from, let's say, the UCSC browser. You don't need to. So whenever you are carrying out a read mapping of some sort of, let's say, SNP analysis or RNA-seq analysis in human, for example, you're not going to do the database this indexing every time when you begin the analysis. You can already download the already indexed files from the internet, for example. So the reason why we are taking this much pain is because the searching becomes much more effective as opposed to the other data structures that we have.
Yeah, sure. Yeah, got it.
Yeah. So this thing, now we have sorted it, and this is what we call as a Burrow-Wheeler transform matrix. You might want to remember this thing. We will come back to this later on. And now all you are doing is you are taking the last column and you're noting down from the top to the bottom what that column is. So what it would read out is GC $AAAC. So remember, we have sorted these rows in lexicographical order, and we are just noting down the last characters of each of these rows and writing them down in the form of a string. This is nothing but the Burrow-Wheeler transform of this particular genome. And this is what usually gets stored whenever you do a compression of your files, that you do like a bzip file or a gzip file. So of course, not in a gzip, but in a bzip, this is what is going to be stored. Now you can further compress it and write it in the form of GC $2A, sorry, it should be 3AC. And this is something what we also call as a run length encoding. So we have created a Burrow-Wheeler transform. Now the idea is that you should be able to go from a Burrow-Wheeler transform and you should be able to retrieve this string back again. Otherwise, there is no point of disrupting this sequence in this particular order. Are you guys clear at this point how we created the Burrow-Wheeler transform?
No, there's a little confusion. Can you repeat the very last part?
Yeah. So what's the confusion? Sorry.
The how you go from BWT matrix to the string.
Yeah, I will come to that. We will discuss that next. But till this point, like creating BWT, you are now. Hello.
Yeah, we lost you for some time. Yeah, it's clear up to that.
Okay. 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 So then after we get this BWT, we are going to discard the rest of the matrix. So we don't need to store that. All you will be storing is this BWT that you get at the end of it. Now, can we retrieve the original genome database G from this BWT? And the answer is yes, because of this property of BWT that is called as a LF mapping property. So let's look at what the LF property is. And L over here stands for the last, and for F stands for the first. So what I have done over here is I have marked the ranks of each of these nucleotides. So for example, if I'm looking at the last row, I look at my G. This is the first G that is appearing in this column, so I write a 1.
Mohak, we lost you at when you said LF mapping property, if you said before something.
Okay, am I audible now?
Yeah.
Okay. We are going to talk about the LF mapping property. So now L stands for last, F stands for first. And what we are going to now do is we are going to assign ranks to the last and the first columns over here. What I mean by rank is whenever I go from top to bottom and I start looking at these nucleotides, what is the nucleotide number that I'm looking at? So when I say that, what I mean is that when I start looking at G, this is the first time that I am seeing a G in this column. So I write 1 over here. Then I move to the next row. This is the first time I'm looking at the C in this particular column. So I write a 1 again. This is the first time I'm looking at a dollar. So I write 1 again over here. Now, this is the first time I'm looking at an A. So I write 1. This is the second time now we are looking at an A. So I write 2. This is the third time I'm looking at an A, I write 3, and so on. I will do the same exercise with the first column. I will write the ranks of these nucleotides as I move from top to bottom. Is the concept of rank clear?
Yes.
Okay. So now there's a very interesting property that you might want to note over here. If you look at this A is the same as this A. So the A with rank 1 in the last column is the same as the A with the rank 1 in the first column. Similarly, A with the rank 2 in the last column is the same as A with the rank 2 in the first column. Are you able to get that? Is this clear?
Sort of.
Sort of. So you can tell me. I can explain it again. So what is it that is not clear? So this is basically what the LF mapping is. So you are able to map the ranks of the nucleotides in the last column to the ranks of the nucleotides in the first column.
Okay.
Yeah. So could you tell me why this mapping property holds true? Why this holds true? Why does it hold true?
Is it because it's a circular permutation of the same string?
Exactly. Because you had done circular permutations is the reason why exactly you will get this property of LF mapping, where you will be able to conserve these positions and you will get the same ranks for a given set of nucleotides. So just remember this property because this is what we are going to take the advantage of in doing the alignments and so on. So let's now start. Let's now move from the Burrow-Wheeler transform to the original string again. Now let's start with, we'll do a cheating as of now. What we are going to do is we are going to take the whole Burrow-Wheeler transform matrix and we are going to dim everything except the last column and the first column in order to understand how you go back. Yeah. So we will start with the G. Can you tell me why will we start with the G? Why will we only start with the G? So remember this last column is a Burrow-Wheeler transform and we want to get to the original string. So how do you know? So remember you will only be given this particular string at the beginning of this exercise, for example. You will not be given any of these matrices or the original string. So how do you know that you will be starting with the G and not with any other nucleotide? Can anybody tell me that?
Sorry, Mohak, I didn't hear this.
So what I'm saying is that we will be now going from the Burrow-Wheeler transform to the original string. So we have to begin somewhere, right? So how do we know that we begin with the G?
I think it will be because when you did the circular permutation,
Yes.
the dollar always goes first, and
Exactly.
you have arranged it in alphabetical order. So,
Yes.
and since your dollar comes first in the alphabetical order, your G will be the last.
Exactly. So that's the thing, right? So the reason why we put a dollar sort of a string was to mark the end of the particular string. So that explains why we are putting something like a dollar in doing these Burrow-Wheeler transform. Exactly what you said. That's completely correct. Because of this, we had put a dollar, and we know that anything behind a dollar would be the last character of that particular genome. Is the reason, and because these are circular, we would then know that okay, we have to start with the G. Yeah, perfect. Does everyone get this? Does everyone get this? Why we are starting with G? Because it's important that you understand it now, otherwise it will be confusion later on.
Yeah.
Cool. So we will start with the G. Now I told you, remember this LF mapping property. So the rank of the G over here would be the same as the rank of the G over here. So that's the reason why you move from this G in the first column, in the last column, to this G in the first column. And then you go back and note what is the last character in this row. Remember, this is a circular permutation, so this C would be coming just before the G. And now you're moving from the left to the right, so you will go back to the C and note down that there is a C behind G. Now, again, you will be able to appreciate the LF mapping property more. Out of these two C's, now which C to choose? Again, you will look at the rank of this C. So the rank of this C is what? What is the rank of this red C?
Two.
Sorry?
2.
2, yes. So the rank of this C is 2. Now we have to find a C in the first column which also has rank 2, which is this particular C. So therefore we know that we have to choose this C and not the one above it. So we move to the C and then note down what is before that C, that is an A, and we note it down. And so the rank of this A over here is 3. So we will move to the rank 3 in the first column and go back, look at what was there before this A, and we will keep repeating this exercise till the end. Now, how do we know that we have to end at this A? Remember that you are going blindly, so you don't know where to end. How do we end at A? I'll give you a hint. It is the same reason why we started with the G.
Has a dollar besides it.
Yes. So because there's a dollar and it's a circular permutation, so you would know that you have to end it over here now. Does everybody get how you moved back from a Wheeler transform to the original string? Does anybody have a doubt? Okay. So remember this we had done, I had given you the first column as well, just to tell you how this LF mapping property works. But what you can also do is you can calculate something called as an LF, which is the left first mapping. So how that would work is, again, you have this last column, you have this first column, and you have the ranks which are associated to these columns. Now, what I have written over here, 1, 2, 3, 4, 5, 6, 7, in the beginning are nothing but the serial numbers. So this is the first row, this is the second row, this is the third row, this is the fourth row, fifth row, sixth row, seventh row. Now what I'm going to calculate is the LF. Because I will calculate this LF, I will not be requiring the matrix, not even the first column over here. So can anybody tell me, if I look at this G in the first column, what is the serial number of this G in the first column? So which row, if looked from the first column, will give me a G, which I am encountering in the last column? Guys?
Seven when it's written there.
Yes. I mean, I'm exactly asking the same thing. So yes, this is the seventh row. When you start looking from the first column, will give me a G, which was there in the first column when looked at from the left side or the last column. So this is, so this will be the seventh, and this C would be the sixth. Sorry, this C would be the fifth because of the LF mapping. This C would be the fifth, dollar would be the first, and so on. So this is exactly what LF mapping would do. But how you are going to calculate is, remember, you will be only given this particular BWT string. So you have to calculate these LFs given this particular string. So how we usually calculate that is by this formula over here. So if I say that I have to calculate, so we want to reach this number 7, but we don't really have this lookup table over here. So how are we going to do that? So count the number of characters which are smaller than G. Can anybody tell me how many characters are smaller than G in any of the columns? So if you take this column, how many characters are smaller than G? And remember, dollar will have the least order. So how many characters are smaller than G in the last column?
3.
Sorry?
Mm.
6, yes. So there are 6 characters which are smaller than the last column, than the G in the last column. And the rank of this G is 1. So you will add 6 plus 1, which is 7. Can you tell me then what would be the LF for C? So in this case, you will be counting the number of alphabets which are smaller than C.
5. 3 plus 2, 5.
Yes, so the answer is 5. So that's the LF of this C. And then you can keep repeating this exercise till the time you get the LF for each and every character in the last column. And now let's just work with this and try to retrieve the Burrow-Wheeler transform. So remember, you will start with the G because, as one of you mentioned, that we always know that we will always be this particular, the first character in the Burrow-Wheeler transform will always correspond to the last in the genome. So we will note down G. Now this G dictates me that go to the 7th row, which is a C. So I'll go to the 7th row and I'll note down that C. Now C tells me that now you have to go to the 6th row. I go to the 6th row, which is an A, and I note down A. Now this tells me that now you have to go to the 4th row. I go to the 4th row and I note down A. Now this dictates me that you have to go to the 2nd row. I go to the 2nd row and I note down C. Then this dictates me that you have to go to the 5th row. I go to the 5th row and I note down A. Now this dictates me you have to go to the 3rd row. I go to the 3rd row, I see a dollar, I stop my search. Do you get this point? Is everybody clear with this?
Yeah.
Cool. So remember, you already only had a Burrow-Wheeler transform. All you will be doing is you will be calculating the LF, and from there on you can find the original string. Now this is for you guys. Will this transform work as effectively for genomes with repetitive regions? So what you can do is you can do the Burrow-Wheeler transform and go back to the original string using this particular string that I've given over here. CAG, CAG, CAG. So only when you do the exercise will you be able to understand this Burrow-Wheeler transform in a better way. So you can try this and you can tell me in the next class probably that if you can get the Burrow-Wheeler transform and back to the original string even for genomes with repeats of such sorts. Yeah. Now let's come to the real deal. Why we all did all of this was to really see if it can also align such things. We are starting with the query, in this case AAC, and we want to find if AAC exists in our Burrow-Wheeler transform GC dollar AAC, which has in turn come from this particular sequence, which is AC AAC G dollar. So again, we will do the cheating first. What we are going to now do is we are coming back to the Burrow-Wheeler matrix, and we start from the 3' end of the read. So this is a read AAC. Let's say this is only 3 nucleotide long, and we want to search if this AAC is present in my genome. So we have this Burrow-Wheeler transform matrix. Now we are going to start with these 2 pointers. One pointer is the top pointer, one pointer is the bottom pointer. And as soon as we look at this C, we will see that where this C is present. So this C is present over here, which is the top, and it is also present in the 6th row. So it's present in the 5th row and the 6th row. So we move our pointers to the 5th and the 6th row, the top pointer at the 5th row and the bottom pointer at the 6th row. We go back, we then look what is the character which is just before the C, which is an A, and even in this case it's an A. So because of the last first mapping property, this is the rank of this A is 2, so we will move to the rank to the A with the rank 2. This A has a rank of 3, so we will move to an A with the rank 3 in the first column, and not this A. So remember, only because of the last first mapping property, we are able to choose out of these 3 which A's do we have to move to. Not this A, but this A and this A, because the ranks of these and these match with each other. So we started with C. Now we move to the A and we get another match over here, which is A. Now you have an AC now. Now we look at the last string over here. It's $1. We will discard this search because we still have my read left to go. And we will look at the other AC, which has an A at the end. We move to this A now, and that's how you know that AAC is located at this particular position in the genome. Are you guys with me so far? Do you understand how we did the query search with using this Burrow-Wheeler transform matrix? Guys?
Mohak, why can't it be the first?
Is this clear? All I'm using is using the last first mapping property throughout. That's all I'm doing. Is this clear?
Someone was asking question. Sorry.
I couldn't hear. Are you asking a question or is it clear?
Abhik was asking a question.
Where is the question?
Yeah.
Is it in the chat?
No. I'm saying it right now. So
Yeah.
why can't it be the first row?
Which one?
In the third and the last figure, you can have AAC on the first row, right? I just have a confusion there. On the first row, above that.
I didn't, sorry.
In the middle.
This one?
Yes.
Oh, remember you don't have this matrix anymore, right?
Mm-hmm.
You only have this particular string.
Okay.
Yes.
Okay, got it.
And also the fact that we are using the last first mapping property, right? So when we look at this C, we have now 2 contenders. So there are 2 contenders for this C. It could be either this C or this C.
Right,
So
right.
you have to.
I got that. Yeah.
Okay,
I got
cool.
that. Yeah.
Cool. Yeah. So then let's try to do this mapping again now without looking at the first column. So again, coming back to the LFs that we already have, we have a T and we have a B. So we have a top pointer, we have a bottom pointer, and we will start off somewhere. Now we will keep shifting our pointers till the time we look at the first C in either direction. So the top pointer comes to the bottom, towards the bottom pointer come towards the beginning. So there you have it. You have located both the C's, the first occurrences of both the C's that were possible. And now you look that the LF of these 2 are 5 and 6. So they're dictating that now you have to go to the 5th and the 6th row. So you move your pointers to the 5th and the 6th row. And if you notice, I have put this thing on the bold over here. So you have located C now already. And you have moved to the next rows. Now it dictates you to go to the 3rd and the 4th row. And also there are A's over here. So definitely you also have a A in your genome, which is a part of the read. You move to the 3rd and the 4th row. Now the third row has a dollar, so you will discard that. You won't continue that search. But the fourth row is a A again, which you note down now. And the fourth row, now this A dictates you have to go to the second row, which is uh, which is a C. But because there was A over here, sorry, this is what you note down in the end, and you move to the last. But now the length of the read has already finished, so you would stop the search at this particular point. Now, what if the read sequence was a TAC instead of a AAC? Remember, till now, if I don't know if you have noticed this or not, we have been only dealing with exact matches. Only if the whole substring matches to the bigger string will be noted. But Burrow-Wheeler transform also takes care of reads which might not be exactly matching in the genome. So for example, in this case, it's a TAC. What would happen is in TAC, for the first time, it would say that there is no TAC in the genome. But it would then do something called as a backtracking. What it would do is it would keep going one step back. And now if you go back at this point, when you had not encountered this A, what it would do at this step is it would try, if let's say it was a TAC instead of a AAC, so you would have matched C and A. But now when you encounter a T over here, what the software would do is it would try other combinations. So it would put a G, C if it matches, it would put a T, it would put A, T, G, C, it would try everything, and it would try to see that if that is what the read has. So when it changes A to a T and is what it matches with this read, it will report the same thing, but also report that there is a mismatch at this particular position. So the solution is backtracking in cases where there are no exact matches. Now, till now, we have only been able to locate, we have only been able to match a read to this Burrow-Wheeler transform or the genome in effect, but we finally want to retrieve the position of this particular read in the genome. So how do we do that? Again, we will do LF mapping. So what the idea now is, so the last time we were at this position, right? So we ended at the second row where you already got an AAC. Now what you would do is you will keep extending this search till the time you reach the dollar sign. So if you have a AAC over here, you will look what is there at the last column, which is a C. Now because the rank of the C is 1, you will then move to the rank C1 in the first column. So you will move to this position and you will note down what is there. It's a A. Then this is the second rank A in the last column. So you will move to the second rank A in the first column. So you will move from this A to this A, and you will encounter a dollar. Hello. Yeah. So you. Yeah. Could I? Should I repeat this again? Hello.
Yeah, it's going fine, I think. Yeah.
Okay. Okay. So then you will encounter a dollar, and that's where you stop. So now we started with an AAC and we went back till the time we reach a dollar. So you will count the number of steps in which you did this. So you took first step, second step, third step. So it took you 3 steps to move from this AAC till the point time you encounter a dollar. So that is what it reports. Finally, that given this genome, your read AAC had a hit in the genome, and it is located at the third position in the genome. So even we will be doing the practicals later on, we will be looking at the output files, which will be reporting only the first position where the read maps. And obviously, given that you know the length of the read, you can always calculate what this last position would be. So the Burrow-Wheeler transform will report the first position where the read had mapped with respect to the genome. Any doubts so far? Are you guys clear? Should I repeat this point again, how we got the position? Is this clear?
I'm okay.
Yeah.
Okay, cool. So then the last part is the RNA-seq experimental design and analysis, and this will be your segue into the RNA-seq analysis, which we will be doing in the next class. So I'll just quickly take you through the protocol once again. So it is almost the same what we saw in the video, except for a few minor changes. So you start with an mRNA or a total RNA, depending on whatever you want to study. Then obviously there will be some contaminant DNA. You would want to remove that, let's say using a DNase enzyme. And then you would select for whatever subsection of RNA that you would want to study in your experiment. So if let's say you want to study only the mRNA, you would want to remove RNA. That usually in, let's say in bacterial RNA analysis, what we do is we use something called as a ribominus, where you remove all the rRNAs and you only then are left with the mRNAs. And then obviously you do a combination of size selection as well. And in case of eukaryotes, if you have to select mRNA, we know that the property of mRNAs in a eukaryote is a poly-A tail, so you can select for that poly-A tail and then select for those mRNAs. But also in eukaryotes, you have long non-coding RNAs, which also can have poly-A tail. So you will then have to use a combination of this selection versus also followed by a size selection. Then you will fragment the RNA into chunks. And finally, you will reverse transcribe this into a cDNA. Can anybody tell me that why can't we go ahead with sequencing the RNAs directly, and why do we need to really convert them into a cDNA? Guys, why are we converting our RNA to a cDNA over here? Why can't we move ahead with the RNA and do the sequencing? That would be much easier, right?
Any guesses?
Okay, because we are in the shortest of time, I'm going to reveal the answer. So RNA is unstable as compared to cDNA, the DNA, right? So during the post-processing, it might get degraded to a much larger extent. So that's the reason why we are converting it into a DNA before proceeding with it. Now, when you make the DNA, finally you put the adapters, as we have already seen in the video. Then we amplify it, and then we select for the sizes, and finally we sequence these ends, and that is what you get in the form of a file. So then it's also worthwhile to discuss what are the kind of questions that you can answer using an RNA-seq protocol. So depending on what sort of thing you want to answer, you have to proceed with the experimental design or the kind of analysis that you will be doing in the end. So let's say one of the things that you can do is a functional study. Let's say, for example, comparing the difference in the gene expression between a wild type or a mutant sample. That's one thing that you can do. So this is what we also call as a differential gene expression analysis. Other thing that you can also do is predict the transcript sequence. So for example, you're interested in any novel transcripts that might be getting expressed in your sample. That is something which you can also do with the RNA-seq analysis. Thirdly, let's say you are interested in studying the mutation in a transcript, but it is not expressing in your sample. So these sort of things you will not be able to answer. So you really need to beforehand understand what is that you really want out of that analysis or the RNA-seq that you are going to do. So therefore, what you want to answer is what decides the experimental design and your analysis strategy. And we will be in the practicals focusing on the first point, which is the comparing the difference in the gene expression of two given different conditions. So choosing which strategy suits your question, you can either have a reference available with you, or you cannot have a reference available with you. So when you don't have a reference available with you, you will have to assemble even the transcripts. So that is what we call as a transcriptome. And because you are not depending on the already available references or the gene models, this will allow you to find any novel transcripts that can occur in your sample. If you were using a reference, then finding any novel transcripts would not have been possible. So depending on what is it that you really want to understand or want to study, your strategy will depend on that. So for our analysis, we will be using this strategy where we will be giving you a reference genome sequence. We will also be giving you the known gene model. So you know that, okay, gene A exists from position X to position Y in this particular reference genome. And we will be giving you the reads. So given all these things, these three things, we are going to carry out the gene expression analysis. So again, this is a read mapping problem. Then finally, how does the data look like? Is your data single end or paired end? So what I mean by single end is that if given this is the fragment or a read, in this particular case, it's a read. So you are only sequencing from one end of the read is what we call as a single end. Paired end would be that you are sequencing from both the ends of this particular fragment, and that's what you are storing. So if let's say you want to do a transcriptome assembly, then because this will give you a better coverage, the paired end read will give you a better coverage, you would want to go for a paired end sequencing. In gene expression studies, it is okay to go for a single end sequencing, but it really depends on even the sub points that you might want to consider. Finally, so the next point is technical replicates versus biological replicates. Technical replicates were more of an artifact when there were no automations that were happening. With the advent of automations, technical replicates are not that much compulsory in today's RNA-seq protocols. But biological replicates, which capture the variation that might exist within the same type of samples, let's say a wild type sample 1 versus wild type sample 2 can still have variation. All those capturings can only happen when you have these biological replicates. Then the length of the sequencing read, again, if you want to do a transcriptome assembly, you might want to get longer lengths of reads as opposed to a gene expression study where you can still have reads towards the lower end of the length. Quality of sequencing reads. So this is something which we are going to talk in depth when we are doing the practicals. Quality also is important. So you might not want to move ahead with the analysis if the quality of the reads that you're getting out of the sequencer are very low. So this might end up giving you false results in your analysis, because these are anyway softwares. So they will produce some sort of a result, but it is up to you to decide if the results make sense or not. So quality is something which we are going to test extensively in the next class, which I think Nitish is going to also discuss what the theory behind it is. Then the next is the coverage. So again, depending on what you want to understand. So let's say if you want to do a transcriptome assembly, you would need a much higher coverage as opposed to a gene expression study. I'll elaborate on this point in the next slide. And finally, PCR duplicates are also something which you might want to look depending on the library complexity. So if let's say there are so what I mean by PCR duplicate is that if I give you a paired end read, and there are, let's say, 1,000 paired end reads which are mapping to the exact same position in the genome, it might be that these are nothing but an amplification bias. But having said that, if they are less in number, you can still think that highly expressed genes might give you, just by probability, these paired end reads mapping to the same exact positions if these are highly expressed genes. So in gene expression studies, you might want to take a call if you want to remove these so-called quote unquote PCR duplicates. And we will expand on this point when we are doing the analysis further. So now gene expression study is a read count problem or a coverage problem. So I will give you an example. If let's say this is your sample RNA and all the highly expressed genes will be having more number of reads that will be contributing in your final sample, as opposed to these all other genes which will be very lowly expressed. So as you can see over here, as I'm increasing the number of reads, 5 million reads, 10 million reads, and so on, only when I reach a higher number of reads am I able to get a good sample of all these other genes. So in a 5 million read, I am getting only a very little amount of all other genes. So therefore, depending on again what the question is, you might want to increase the number of reads and thereby the coverage of your reads that you are trying to look at. So gene expression analysis is more of a read count problem than a coverage problem. It does not matter if your entire gene is getting covered by all the reads, but if you are able to get some coverage even to some position in the read, you might also want to consider that. So it's a read count problem. So what I mean by that is that there is a wild type and a mutant. This blue bar over here is the genome of the wild type, and this red bar over here is the mutant of the genome of the mutant, and these arrows they mark the position of the beginning of the gene A, B, and C in respective genomes. So these black bars will represent the RNA which is coming out of this this particular genome. So what it seems is that the genome in wild type, the gene B is not expressed at all, but in mutant the gene B is somewhat expressed. The gene A in wild type is highly expressed, but the expression of gene A becomes very low in the mutant. And this is all what we are going to do in the analysis. We are going to compare these read counts, which will be our readout for the expression of genes in our genome. So finally, to end it all, to move towards the beginning of the analysis, what we are going to finally see in the next class is, given that you have a reference genome sequence and a short sequencing read and a gene model for all these for the genome, you will pass them through a particular black box, such that at the end of it you get the differentially expressed genes. So our motive in the next coming classes or the analysis classes would be to understand this particular black box. How does it calculate the differentially expressed genes? Yeah, so I'll end the session. I will be taking any questions that you have with respect to the talk. Also, before ending, I would really encourage that you go back and do that Burrow-Wheeler transform exercise for that CAG repeat genome that we had discussed. Thanks.
So if there are no questions, I would like you to do some amount of homework for next session. Okay, so yeah, so we will be going through we will be starting from like when we get reads of any NGS like specifically RNA-seq data, and we would be going through the starting from the quality mapping and all the things. I will tell in between, like in between the steps. Yeah. Am I audible?
Your voice is cutting off.
I think you got cut off in between.
Yeah, I'll repeat. Yeah. Okay. So I would like you to do a homework on. Yeah. Am I audible right now? Yeah. Sure. Hello.
You're audible. You're audible.
Yeah, okay. If my voice breaks, please let me. Yeah. Okay. So I would like you to do homework on like what are the application of RNA-seq data other than differential gene expression analysis. Okay. And such that I will tell you how those applications can be achieved, like how those things can be achieved in between the processes that I will tell you. Second, what do you mean by quality of any read data? Like any guesses, or like any educated guesses, like you can search internet or do whatever. So I would just would like you to, I will ask these two questions before starting the Tuesday session. So please note it down, and please like do your homework, and that's it. Yeah.
Hey, can you summarize the two questions again?
So first question is, what are other applications of RNA-seq data other than differential gene expression analysis? Like, right, other than differential gene expression analysis, or is it only differential gene expression analysis that you can do from RNA-seq data? Okay. And second question is, what do you mean by quality of reads? So as Mohak mentioned, like you need to check the quality of reads. What do you mean by that? Okay, so that's it. These two questions. These are two bit vague questions, but I would like you to brainstorm on these two 2 questions. Yeah.
Okay.